Hi class, and welcome to this screencast on evolution. So we're going to go ahead and move forward on our topic of evolution and focus on evolution of populations. And so this screencast is called the hardy-weinberg principle, and we're going to see why here in just a second. So the main question that I want you to keep in the back of your mind as we go through this screencast, because it's going to get pretty detailed today, is how do we know a population is evolving? So I want you to try to come up and think of things on your own right now. How do we know a population is evolving? You can probably come up with a multitude of answers, right? There are many different fields of science that we can look to see if a population is evolving. We could look at a fossil record. We could look at the anatomy and physiology of them. We could look at the biochemistry, the molecules, the DNA to see if it's evolving. So one way, and the way that we're going to look at it today to see if a population is evolving, is to determine what the genetic makeup of a population would be if that population were not evolving. I know this might seem counterintuitive right now, but so then what we would do with that, we would find what that number is, okay, we would find what the genetic makeup is, if it hypothetically were not evolving, and then we would compare this number with data from a real population. So then, if there are no differences between this hypothetical not evolving data, and there were no differences between that and the real data, then what would you assume? If you said that the real data is not evolving, you are correct. But if we do compare this real population, this real data, to our hypothetical numbers, and there are differences, then that tells you that yes, the population is in fact evolving, because that genetic makeup, those numbers, are different than what we had predicted if it was not evolving. I know that sounds really confusing, and I hope that I can clear things up as we go. So, how do we do this? How do we determine the genetic makeup of a population if it were not evolving? We use something called the hardy-weinberg principle. This describes the gene pool of a population that is not evolving. Now, there are 5 conditions that must be met in order for a population to be considered not evolving. And those 5, and you have to know these 5. No mutations can be happening. Mating must be completely random, because remember from our previous screencast, if there is any selection going on, any sexual selection, that's going to be adaptive, that's going to be a type of natural selection, and evolution is going to occur. No natural selection, obviously, that's the driving mechanism for evolution. An extremely large population size. Remember, we sort of looked at this with the bottleneck effect and the founder effect, that evolution is sort of magnified in these small populations. So we want to make sure the population is very large. And no gene flow, right? No immigration or emigration of genes or alleles into or out of a population, because that can also cause evolution. So all of these things, all 5 of these things, we've already discussed. We discussed in the first 2 screencasts. All of these are going to lead to evolution. The biggest one is natural selection. That's the one that's adaptive. And then large population, gene flow, mutation, these are things that are sort of non-adaptive, right? These random chance processes. Okay, here's the catch though. These 5 conditions are almost never met in nature for an entire population. You could probably not name me a population that has all 5 of these conditions met. So you might be thinking, well, what's the point? Why do we use this principle if these conditions are never met in a real population? Well, the answer is that specific traits can actually be fixed in a population. When I say fixed, I mean not evolving. So we can use this principle, probably not on entire populations, but we can use this principle on specific traits. And so that's what we're going to do for the rest of this screencast. Okay, so let's just go ahead and assume that all 5 conditions that we just stated are met for a given gene. And we're going to take a look at this single gene, and in our hypothetical situation, we're going to look at flower color and a population of wildflowers. Okay, our population of wildflowers has 500 flowers, so there's a total of 1,000 alleles. Because for each gene, that flower has 2 alleles. It got 1 allele from mom, or one parent, and the other allele from dad, or the other parent. So each gene has 2 alleles, so 500 times 2 alleles for each of those genes is a total of 1,000 alleles. Now let's take a look down here at these numbers here. We have 800 red alleles and we have 200 white alleles, which basically means we have 400 red flowers, right? And we have 100 white flowers. But we're going to count alleles. Remember, allele is just simply a version of a gene. It's either red or it's white. Now, let's draw our attention to this over here now. This is part of our hardy-weinberg principle. We're just going to go ahead and call this red allele, it's dominant in the population, and we're going to call that P, and white is recessive in the population, and we're going to call that Q. Now, P plus Q equals 1. 1 is the total amount of wildflowers in that population, which is really 500, right? We're just going to simplify it and use 1. So all the P's, all the red alleles, plus all the Q's equals 1, the total amount. Now, let's draw our attention to this last bullet point. Assuming random gamete formation, which gametes are just sperm and egg, there is an 80% chance of a red or egg sperm because there's 800 red alleles, so 80% chance that an egg or a sperm will be red, and only a 20% chance, because of this 200 number here, only a 20% chance that that egg or sperm is going to have a white allele. So hopefully that makes sense so far. But the question that we need to keep coming back to is, okay, what's the point? How is this gene inherited in a population? Is this gene, is this allele evolving in a population, or is it not evolving? So now to answer that question, we have to calculate fertilization probabilities. We have to take these percentages down here of the 80% and the 20%, and we have to calculate probabilities of them fertilizing one another. So we have to do that one by one. So let's start with red gametes. The probability of 2 red gametes coming together is 0.8 times 0.8. Why 0.8? Because of this 80% number down here. 80% chance of a red sperm. Okay, so 0.8 times 0.8 is going to give us 64% chance that we'll have 2 red gametes. Same idea going with 2 white gametes. It's that 20% times the 20%. So 0.2 times 0.2, and that's a 4% chance. Now we have this last possibility, which is kind of interesting. It could be a combination of a red and a sperm and a white egg, or a white sperm and a red egg, right? So how do we configure that? Well, we have to have a 2 in front of this now. 2 times the red times the white is equal to 32%. So keep all of these in mind. We have this, the red, which remember, going back to this slide here, that is P. Dominant is P, recessive is Q. So this is really P squared, P times P. This is really Q squared, isn't it? And this is 2 times P times Q. So hopefully you followed me this far. So here's our finale. The number of all of these alleles, remember, must equal 1, or 100%. So the final equation for the hardy-weinberg equilibrium, when the gene is fixed or not evolving, is P squared plus 2PQ plus Q squared. We basically just added up those 3 fertilization probabilities that we just calculated on the previous slide. So red times red, white times white, or it could be 2 possibilities of the red-white combo, and that's this here. Now, what's really important to understand is that because red is dominant, these 2 numbers here actually represent the dominant phenotype of red. If you are P squared, you're red. If you're 2PQ, you're red. Now, the only way you're going to be white is if you're Q squared. Okay, so this is the recessive phenotype. Yes, we are using math. Okay, so this is bringing it all together, I hope. We're using math to calculate changes in allelic frequencies and thus providing evidence that evolution is or is not occurring. And I'll come back to that in a second. Finale, again, part 2. I really want to make sure you understand this. This law essentially states that if no evolution is occurring, this should say if here, if no evolution is occurring, then an equilibrium of allele frequencies will remain in effect in each successive generation of sexually reproducing individuals. So these frequencies that we would calculate here, whatever these would be, would stay the same if evolution was not occurring. If they start to change, okay, in a real data set and real nature, then we know evolution is occurring. We're comparing it with this hardy-weinberg equation. Okay, so this law is only good, like I just said, when it is applied to real-world problems. So for example, we have observed versus predictive frequencies of alleles. So the hardy-weinberg calculation gave us these predictions, and then some scientists went out in the field and actually counted these observed frequencies. And so when you compare the two numbers, are they the same? Well, obviously, no, they're not. And so this trait, whatever it was, whatever they're looking at, is evolving in this population. Okay, just some terminology as we do some practice problems. Allelic frequencies are going to be just your P and your Q. Remember, P is dominant, Q is recessive. This is genotype, the genetic makeup, not what you see. The phenotype is your P skewed, your P squared, your 2PQ, or your Q squared. So remember, these two are going to show the dominant phenotype, what you see, so the red, and this one is going to show the recessive phenotype, which would be the white in the case of our wildflowers. So this is phenotype, and these two are genotype. So let's do two quick practice problems, and then I'll give you one to do on your own. Okay, so in the human population, the recessive PKU gene is fixed. It's not evolving. This gene makes an enzyme which helps humans break down a certain sugar, and if you have this PKU recessive gene, you can't drink things like Coke and Diet Coke. It's actually written on the little side of the label. We know from our data sets when we do our testing at birth that the occurrence of this recessive PKU gene is 1 in 10,000 births. Okay. What is this number? That's what we have to first figure out. Let's not even worry about what they want us to calculate. What is 1 in 10,000 births? Is this a genotype or is it a phenotype? Hopefully you said it's a phenotype, okay? So 1 in 10,000 is going to be the phenotype Q squared. So 1 over, and I'm using my mouse, so I'm sorry, that's Q squared. So what do we do with Q squared? Well, let's just do what we can with it. We can actually find Q from this, right? So we can take the square root of 1 over 10,000, and that equals 0.01. Okay, what do we do with that? Well, let's go back to our very first equation we learned, which was P plus Q equals 1. We know Q, so all we have to do is solve for P, so P equals 0.99. Okay, we've done pretty good so far. We have Q and we have P. Now let's see what they want us to calculate. They want us to calculate the percentage of heterozygotes in the population. Well, what would be a heterozygote? A heterozygote would be this 2PQ. Remember that red-white combo that we looked at. So now we just have to do 2 times 0.99 times 0.01, and that equals 0.0198. But that's not what they asked for. They asked for the percentage. So that percentage is 1.98%, or about 2%. So that's how you do the Hardy-Weinberg equation. Let's do one more really quickly. We've got a population of butterflies. Brown is dominant over white. A 1940 study showed that 40% of all butterflies are white. So again, before we even see what they want us to calculate, let's worry about this number here that they give us. What is this? What is 40% of all butterflies are white? Yes, it's a phenotype, and it's the recessive phenotype. Very good. So 0.4 is equal to Q squared. This is sort of a trend in Hardy-Weinberg problems where you're given this information to begin with, and then you go ahead and you find Q by taking the square root, and Q equals 0.63. Again, using our same beginning equation, P plus Q equals 1, we find P, and P is equal to 0.37. So now they want us to calculate the percentage of heterozygous butterflies. So remember, this is 2PQ. So 2 times P times Q is equal to 0.46, but they ask for a percentage, which is 46%. And the frequency of homozygous dominant, this would be P squared, which is equal to 14%. Now I want you to read this last question on your own, and I want you to answer that in your notes. And I want you to answer this next question in your notes as well on your own. And we'll talk about all of this and do lots more examples, I promise, in class.